Hi everyone. My name is Eryk Kropiwnicki, and I'm a bioinformatician in the Mion Lab, and I'll be talking about Drugshot. Let's get into it. So Drugshot is an Aperture and a standalone web application that allows users to query biomedical terms of interest to receive a list of prioritized small molecules related to their query term. And so these query terms are sent to the PubMed API, and a list of PubMed IDs associated with the query term are retrieved. Drugshot then outputs a prioritized list of small molecules associated with this query term based on co-mentions between PubMed IDs that include both the drug and our query term. And so then using the top drugs from this, what we'll term associated list of small molecules, Drugshot further predicts additional compounds based on literature co-occurrence and signature similarity from drug-drug similarity matrices, which I'll sort of discuss more broadly in a subsequent slide. And so Riff and Autoriff are the underlying database of drug to PubMed ID associations for Drugshot. And so we sort of compiled this master list of compounds and queried them using various existing APIs. And so Riff contains drug to PubMed ID associations that were manually curated by the NCBI's PubChem portal. And Autoriff is an automatically generated drug to PubMed ID association resource that we created by querying the PubMed API with unique drug terms. And so sort of the distinction here is that Autoriff is a more comprehensive resource, albeit it may be more prone to errors because it relies on fuzzier string matching to retrieve PubMed ID associations automatically versus Riff, which is more curated, so to speak. And just to sort of give like a visual for how this Drugshot search is conducted. Again, we have this query term that's chosen by the user, which is submitted to the NCBI eUtilities eSearch API, which we just termed the PubMed API. And we find PubMed IDs of abstracts that mention this query term. And then the PubMed IDs from this search are compared with the drug to PubMed ID associations found within Riff or Autoriff, which are like pre-computed and can be chosen by the user. And lastly, we have a table of drugs associated with the query term that's produced, which is ranked by the number of co-mentioned PubMed IDs between the term and the drug. So coming back to these drug-drug similarity matrices, there are two types that are used for predicting further compounds. One of which is the literature co-mentions matrices created from Riff and Autoriff, which contain pairwise counts of shared PubMed IDs between all compounds in the respective association file. So there's two separate matrices for literature co-mentions. And then we also have this sort of more interesting signature similarity matrix, which contains pairwise cosine similarity of drug-induced L1000 gene expression signatures for approved and experimental drugs from the LINKS project. And so the top predicted compounds from the chosen matrix are ranked by the largest mean similarity values with the compounds in our associated drug set, which is sort of like a reference set from the literature associated with our query term. So now I'll kind of just give a practical example of this and dive into a demo of the standalone website. So here is the PubMed query page or the homepage for Drugshot. And so we have this search bar that allows users to query terms that they want to include in the search and terms to omit from the search. On the right, we have a input field for selecting our unweighted drug set size, which is then used to predict the additional compounds from the chosen similarity matrix. By default, this is the L1000 signature similarity matrix. And there's also a toggle for switching between Riff and Autoriff. So for this search, we're querying the example term lung cancer here and using Autoriff as sort of our background drug to PubMed ID association file. And so I should also mention this is like very much an offshoot of GeneShot down to the design of the website. And you'll see similar figures here as Alex had shown before, which is the scatter plot, where the x-axis plots the publications that co-mention a search term and the drugs. And then on the y-axis, we have the sort of normalized fraction of publications, where we look at essentially the specificity of each drug to each term. So anything that's sort of higher up, those higher up points are more specific to lung cancer. And you can mouse over these points. So cisplatin would sort of be an example of a compound that's co-mentioned a lot with lung cancer, but isn't necessarily specific to lung cancer. And so we could scroll down to these two tables. On the left, we have this associated compound table. So these are the top drugs from literature. And then on the right, we have the drugs associated by drug similarity. And so in this case, it's the L1000 signature similarity matrix. And these are the top compounds that are sort of related to our unweighted drug set of drugs associated with lung cancer. And these tables can be downloaded, and there's also a way to submit them for drug set enrichment analysis, where users can sort of look at common genes, side effects, pathways, etc., that the drugs may share. And so we also have a drug set augmentation feature where users can submit their own drugs and select a similarity matrix of their choice. And again, based on our unweighted drug set, we get a ranked list of compounds ranked by their cosine similarity of their drug-induced gene expression profiles to this unweighted drug set. And lastly, this is the Drugshot Aperture. Apertures will sort of be discussed more in depth by Daniel and Sherry later, but this is sort of Drugshot in a different form. A lot of the same input options here are available. And once the user populates these fields and submits the form, a Jupyter notebook is executed in the cloud. In this case, I've already executed this specific set of input form options. So we have a cached notebook here of the results. It's a persistent report. And at the top, there are ways to toggle the code, run the notebook locally, or just download the notebook. As we scroll through the notebook, we see a lot of the same figures. For example, this table of associated compounds from the literature. If we scroll down further, we have our scatter plot, a nice plotly plot here with mouseable points. And you can also sort of click on the table of contents to navigate between these sections. The Aperture exclusively also contains this ROC curve for the unweighted drug set rankings in the matrix, which sort of gives you a metric of how relevant the predicted compounds may be based on how well the prediction matrix was able to retrieve the true positives, let's say, associated with lung cancer. So that gives us some confidence that our predictions have some validity. Of course, we have a downloadable table of the top predicted compounds from L1000 signature similarity. And lastly, bar plots of drug enricher analysis of these predicted drugs, which include L1000, or sorry, Go biological processes and side effects from CIDR. And there's also a link to a more complete report. So lastly, I'll wrap up here and talk about some conclusions and future directions. So hopefully I've shown that Drugshot is able to rank compounds associated with any queried biomedical term using PubMed. And we're able to prioritize other compounds that may be associated with the query term based on these drug-drug similarity matrices. We can use Drugshot to create novel drug set libraries that can be used to prioritize compounds for drug repurposing opportunities. We're looking to update Drugriff and Autoriff, as well as the signature similarity matrix to support larger compound pool and incorporate the latest release of the Lynx L1000 data. And in the future, we also plan to develop libraries of drug to disease associations from Drugshot with predictions that can be ready for experimental validation. And that's actually something we're currently working with collaborators at NYU to validate compounds for atherosclerosis. So that wraps it up. I'd like to, of course, thank everybody in the lab and the bolded people who helped with the project. And I'll take any questions in the chat.